Every second, millions of cells in your body divide, but not all divisions are the same. Some create identical copies, while others create you. There are two types of cell division, mitosis and meiosis. Mitosis is for growth and repair. Meiosis is for reproduction and genetic diversity. Mitosis produces two identical diploid cells. Meiosis produces four non-identical haploid cells. One division versus two divisions. No variation versus high variation. Before division begins, the cell enters the S phase, where DNA doubles. But here's the key: chromosome number stays the same, only DNA content increases. Same chromosomes, double DNA. Mitosis happens in 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase. Chromatin condenses into visible chromosomes. Centrioles move apart. Spindle fibers begin forming. And the nuclear membrane breaks down. Chromosomes align at the equator, the metaphase plate. Each chromatid attaches to spindle fibers from opposite poles. Cohesin proteins break. Sister chromatids separate and move to opposite poles. New nuclei form. Chromosomes relax. And the cell splits into 2 identical daughter cells. Cancer drugs like vinblastine and Taxol stop mitosis by freezing cells in metaphase. This is mitosis under a microscope. The blue structures are chromosomes, and right now, they're lined up in the center. This stage is called metaphase. Next, the green spindle fibers pull them apart to opposite sides. That's anaphase. As the chromosomes reach the ends, the cell prepares to split, late anaphase. In just a few moments, one cell becomes 2 identical cells. That's mitosis, the process that helps your body grow and repair. Now we reach the final stage of mitosis, telophase and cytokinesis, the grand finale. At this point, the chromosomes have already reached opposite ends of the cell. They begin to relax and uncoil, returning to their normal chromatin form. Around each set of chromosomes, a new nuclear envelope starts forming, creating 2 separate nuclei. The nucleolus also reappears, indicating that the cell is preparing to resume its normal functions. But the division is not complete yet. Now comes cytokinesis, the actual splitting of the cell. A contractile ring made of actin and myosin forms at the center of the cell and tightens like a belt, pulling the cell membrane inward to form a cleavage furrow. This furrow deepens until the cell is completely divided into 2 separate daughter cells. In the end, one parent cell becomes 2 genetically identical diploid cells, each ready to enter interphase and continue the cycle of life. Now let's move to meiosis, a specialized type of cell division that creates reproductive cells like sperm and eggs. The main purpose of meiosis is to reduce the chromosome number from diploid to haploid, so that when fertilization occurs, the normal diploid number is restored. But meiosis does more than just reduce numbers, it also creates genetic diversity. Unlike mitosis, meiosis involves 2 consecutive divisions, meiosis 1 and meiosis 2. But importantly, there is no DNA replication between them. As a result, one starting cell ultimately produces 4 haploid cells. A key feature of meiosis is genetic recombination, where homologous chromosomes exchange segments of DNA during early stages. This process, known as crossing over, generates new combinations of genes, making each gamete genetically unique. There are also important differences between males and females. In females, meiosis begins before birth and can remain arrested for many years, while in males it starts at puberty and continues throughout life. Additionally, female meiosis produces 1 functional egg and smaller polar bodies, whereas male meiosis produces 4 functional sperm cells. Clinically, errors in meiosis, especially non-disjunction during meiosis 1, can lead to chromosomal abnormalities such as Down syndrome, and the risk increases significantly with maternal age. So in summary, meiosis is not just about division, it is the process that ensures variation, reproduction, and continuity of life. Now we enter the most complex and important phase of meiosis, prophase 1, which is divided into 5 substages, each with a specific role in genetic variation. It begins with leptotene, where chromosomes start condensing and become visible as thin threads, while homologous chromosomes begin to search for each other. Next is zygotene, where these homologous chromosomes pair up in a process called synapsis, forming a tight alignment with the help of a structure known as the synaptonemal complex. Then comes pachytene, the most crucial stage, where synapsis is complete and crossing over occurs. Segments of DNA are exchanged between homologous chromosomes, creating new genetic combinations. After this, in diplotene, the synaptonemal complex breaks down and the homologous chromosomes begin to separate, but they remain connected at points called chiasmata, which are visible evidence of crossing over. Interestingly, in females, meiosis can remain arrested at this stage for many years. Finally, diakinesis marks the transition toward division, where chromosomes condense even further, the nuclear membrane breaks down, and the spindle apparatus begins to form. Together, these 5 substages ensure proper pairing, recombination, and preparation for the reductional division of meiosis, making prophase 1 the key to genetic diversity. Now let's understand how meiosis is completed through 2 consecutive divisions, meiosis 1 and meiosis 2. Meiosis 1 is called the reductional division because it reduces the chromosome number from diploid to haploid. During metaphase 1, homologous chromosome pairs, known as bivalents, align at the center of the cell, and their orientation is random, which contributes to genetic variation. In anaphase 1, these homologous chromosomes are pulled apart to opposite poles, but the sister chromatids remain attached. This is the key difference from mitosis. As a result, each daughter cell receives only 1 chromosome from each pair, effectively having the chromosome number. Following this, the cell quickly enters meiosis 2 without any DNA replication. Meiosis 2 is similar to mitosis, where the sister chromatids finally separate. The centromeres divide, and chromatids move to opposite poles, resulting in 4 haploid cells. However, unlike mitosis, these cells are genetically different due to the earlier crossing over and independent assortment. In males, all 4 cells develop into sperm, while in females, the division is unequal, producing 1 functional egg and smaller polar bodies. So overall, meiosis 1 reduces the chromosome number, and meiosis 2 separates the chromatids, together producing genetically unique haploid cells essential for sexual reproduction. Let's connect all of this to real clinical importance. Certain cancer chemotherapy drugs, like vinblastine and paclitaxel, specifically target the mitotic spindle. By disrupting microtubules, they arrest cells in metaphase, preventing cell division, which is particularly effective against rapidly dividing cancer cells. On the other hand, ionicing radiation can damage dividing cells by inhibiting mitosis and causing errors like non-disjunction. This is why tissues with high turnover, such as skin and mucous membranes, are especially vulnerable, often leading to ulceration. One of the most important clinical outcomes of meiotic errors is Down syndrome, or trisomy 21, which occurs due to non-disjunction, most commonly during maternal meiosis 1. The risk of this condition increases significantly with maternal age. In fact, many chromosomal abnormalities are incompatible with life and result in spontaneous abortion. Additionally, defects in processes like synapsis, crossing over, or spindle formation during meiosis can lead to infertility or recurrent pregnancy loss. So in summary, proper cell division is not just a basic biological process, it is critical for growth, reproduction, and overall health, and even small errors can have major clinical consequences. Finally, let's bring everything together with a quick comparison between mitosis and meiosis. Mitosis is a single division process that produces 2 genetically identical diploid cells, and it mainly occurs in somatic cells for growth, repair, and maintenance of the body. There is no genetic variation here. The daughter cells are exact copies of the parent cell. In contrast, meiosis consists of 2 consecutive divisions and produces 4 haploid cells, each genetically unique. It occurs in the gonads to form gametes, and it introduces variation through processes like crossing over and independent assortment. Another key difference is in chromosome behavior. In mitosis, sister chromatids separate, whereas in meiosis, homologous chromosomes separate during the first division, and sister chromatids separate during the second. So, in simple terms, mitosis maintains stability by producing identical cells, while meiosis creates diversity and prepares cells for sexual reproduction. Understanding this difference is fundamental to grasping how the human body grows, repairs itself, and passes genetic information to the next generation. If you found this explanation helpful and easy to understand, make sure to like, subscribe, and share for more high-yield medical content. Keep learning, keep growing, and I'll see you in the next video.